4 shots are fired, 4 animals are hit. The anaesthetic takes a few minutes to work. To take the samples, the team needs to act quickly. The animal is placed in a position which ensures it won't suffocate.
9 years.
Why did you look at the mouth?
To know the age. This female is about 9 years old.
The team take blood samples which will be tested for diseases such as Rift Valley fever, bovine tuberculosis and brucellosis.
Green one. Green one.
What are you doing?
Collecting a little bit, a piece, a small piece of ear for genetic material to study the genetic variability and the DNA of the buffalo. This is a smear to detect blood parasites from the buffalo, mainly transmitted by ticks, tick-borne diseases.
Fecal samples are also taken.
Fecal sample for egg count.
Did you take the swab?
Yeah.
Okay.
Anybody on the radio there?
Yes.
Yeah, please, I can hear you.
We almost.
Okay, can we drop the next 3?
Yeah, we are just about to reverse this one. We left the other one to this other group. So
The tested animals are marked so they won't be targeted again. The paint should only last a few days and should not affect the animal's relations with others in the herd. Details of the animal's age, size and sex are also taken to keep with the samples. The location where the animal was tested is also recorded. The samples are put in an icebox.
So this is the antidote that we're using. It's the drug used to reverse the anesthetic that the animal received.
The product used to reverse the anaesthetic is naltrexone. It only takes a few minutes to work. Three sampling teams work at the same time.
We are taking a chip of the ear for DNA testing for us to identify the animals and check how related they are. We are also taking blood in 3 different types of tubes for different tests which require different states of the blood. We are also taking a thin smear of blood on microscope slides to check for blood parasites. There is also a probing sample which we are taking, for example, in this one, in animals which are younger, for foot and mouth disease virus.
We are taking some probings The probants are taken from the larynx, whereby you scrape and then preserve so that you can find the virus in there. So that is the main sample we are taking for FMD in this exercise. After preparing the samples, we put them in this big can here, which has liquid nitrogen at minus 178 degrees, and it will make our samples viable until we get to the lab. The liquid nitrogen we have in this can last us up to a month. Yeah, so we still have quite a lot inside. So I finished with our sample preservation.
In 2 weeks, 180 animals were tested in total, half in the Chobe National Park and half here in the Okavango. The last group tested are the lucky ones. They don't need the orange mark on their backsides.
This is the drop of blood that we used earlier to make a slide to look for blood parasites. And we're dipping it in alcohol so that the blood cells are fixed and they don't get damaged. And now we put the slides with toilet paper very carefully so that they don't get damaged. And then we put them in an envelope like this one.
This kind of survey helps us in that it gives us a picture of the disease status of our wildlife, and it should be, and we hope it will be, an ongoing process that can be done.
Since I'm now experiencing a lot of things, the challenges on sampling buffaloes, and also at the end I will help the department, or the department will benefit from the experience I got in this project.
They have to be done within 8 hours after sampling. So I hope you are still within time.
Time is short. The samples need to reach the laboratory within 8 hours. The helicopter takes the samples to the nearest airstrip, and then the plane takes them to the laboratory of Mourn's Lesholathebi Memorial Hospital to be treated.
He will come to the airport to collect the box, and we will take it to the laboratories, and we will then process the sample. Okay?
Finally, the samples will be sent to different laboratories which are part of an interregional partnership, such as the Agricultural Research Council's Onderstepoort Veterinary Institute in South Africa and the Botswana National Veterinary Laboratory in Gaborone. Its technical expertise on transboundary animal diseases allowed the FAO ECTAD to coordinate the national teams and the international experts in joint action and capacity building. Such operations should continue in order to identify and prevent animal diseases and ensure health for wildlife, cattle, and the human populations in these regions.
take